Good afternoon. Firstly, I would like to thank you for taking your time and joining to my presentation. Today, I'll talk about my research activities and which are mainly focused on controlled environment agriculture. And briefly about my background, I have a PhD in Sabancı University from plant nutrition and plant physiology. Then I started working as a research assistant at ETH Zurich in the group of plant nutrition in a cost project. There, we investigated the bioavailability of zinc in humans, which was hydroponically grown and biofortified with 67 zinc isotope. Then I became a postdoc at ETH Zurich in a European Space Agency project in the Melissa food characterization phase 2, cultivar selection. And then in that project, we investigated the microbial community dynamics and response to plant growth, promoting microorganisms of food crops. In parallel, I was working in an automated multi-sensor system for plant crop phenotyping. And the target plant was ryegrass. Ryegrass is an important crop in Switzerland because Swiss are proud of their cheese and for their milk, and ryegrass is counted as a crop. And there my role was to correlate the QTS, the breeding traits that they are interested in 250 European elite ryegrass cultivars, to correlate these traits with the higher accumulation of micro and micronutrients, which are important to increase the yield of ryegrass. And in my second postdoc project at European Space Agency at ETH Zurich, that was a project from the Melissa Foundation. There we investigated the wheat root plasticity and allometric root-shoot relationships under 7 different macro and micronutrient deficiencies in different durations of deficiency conditions in hydroponics. After working at ETH for 5.5 years, I got a job in Earth Space Technical Ecosystem Enterprises in Switzerland as a food production project manager. The project was connected and controlled indoor vertical farming of aeroponics, hydroponics, and ebb and flow. In my last job position, I worked at Green City Solutions in Germany as a mouse farm manager. The company was designing, producing, and selling an outdoor botanical air biofilter to improve the ambient air quality in highly polluted regions in the city. And now, very recently, I am granted with a co-funded European Union Horizon 2020 Marie Curie Actions project with TÜBİTAK 2236, and I am working as a principal investigator. In the agenda of my talk today, I will mention about these 2 projects at ETH Zurich and the vertical farm in Lausanne, and the city tree product in Berlin, and my current project at Sunum at Sabancı University. So in the first project conducted for European Space Agency at ETH Zurich, we investigated the microbial community dynamics and response to PGPM in the rhizosphere of 4 crops cultivated in hydroponics. Melissa Foundation is a space research program in European Space Agency. It has 4 compartments, including the microorganisms and higher plant-based ecosystem. It's a 70% closed-loop ecosystem. And European Space Agency's interest is the use of these hydroponically grown plants and microbes to use them for food production and for wastewater processing, for wastewater treating and polishing for recycling applications. Therefore, it's important for Melissa program to establish reliable and robust microbial communities in the higher plant compartment to assure a sustainable food production and to use them for wastewater recycling using the plant. And in that project, Ghent University was responsible for the potato experiments, Guelph University was for durum wheat, and Napoli University was for soybean, and we were responsible to conduct the experiments with bread wheat. In the framework of this project, aimed at the development of biological life support systems for manned missions in space. Therefore, we investigated the effects of these PGPMs, these plant growth promoting microorganisms, on 4 common crops. These are selected crops of ESA, bread and durum wheat, soybean and potato. And these are cultivated in recirculating hydroponic system for the whole life cycle. And each crop was inoculated with a commercial PGPM mixture that was purchased. And the composition and the dynamics of these microbial taxa, this microbial community embedded in the root rhizosphere and in the nutrient solution in this recirculating system was characterized through 16S and ITS targeted Illumina MySeq sequencing. And the ETH plant wheat experiments was carried out in a convirant growth chamber. And the tests were conducted under very clean system, almost aseptic conditions, to be able to analyze and to see the effect of the inoculated microbial inoculation with preventing as much as contamination from the outside microbes, airborne pathogens as well. And there we used a commercial microbial mixture for all plant species, which was including 48 microbial taxa, and it's called Myco madness. These are some webcam captures from the growth chamber. Since the plants were growing under aseptic conditions, the entry in the walk-in growth chamber was restricted. Hence, the plant development was followed up by a webcam installed inside the chamber. And in this picture, you can see from seed to seed generation when the wheat is very small and when it is almost flowering, and you can see the spikes. Now I will show you the microbial community dynamics and plant responses to plant growth promoting rhizobacteria. On the left graph, you can see 4 different crops and how this inoculation, this Myco madness inoculation, was investigated on the plant root zone. I won't go very detailed on each microbial taxa here, but in general, bread, wheat, and potato, each of these 48 microbial taxa isolated from Myco madness was represented. In soybean and bread wheat, the level of investigation of the root zone by Myco madness microbial taxa was higher. You can also see the gray labeled columns of these microbes are isolated from Myco madness, and the control plants, these are the white labeled columns. And interestingly, potato, it had maybe a problem in terms of contamination in the control plants, because the diversity of the microbial communities of this root rhizosphere was higher in the control plants than the Myco madness inoculated. And I will just show you I would like just to show you the bread wheat rhizosphere response to this commercial mixture. On the upper part of the graph, you can see 18 weeks of the desinutrient solution analysis. On the upper part, that's the control group, which there was no inoculation, just started almost sterile conditions. And in this control group, you can see that at the first 2 weeks, rhodospiralis was dominant. And after the second week, there was a drastic change. There was a sharp change from rhodospiralis to cyanobacteria. And the cyanobacteria stayed in the system till week 10th. And at 12th of the week, again, there was a dominance of rhodospiralis. And then this all bacterial microbial dominance was converted to fungi at the end. And these nutrient solutions were sampled every 2 weeks to analyze them with immunomycete sequencing. And at the down part of the graph, that these are the Myco madness inoculated plants. There, it started first with fungi dominance, and it was partly shifting and replacing each other with rhodospiralis and fungi. And by the 12th week, the cyanobacteria became dominant, and it just drastically changed again, and with fungi. That was interesting finding out in the experiment too, because the inoculation composition that we purchased, this Myco madness, to assess them wasn't one of the aim of the study, but its microbial taxa was remarkably different than its packing. One of the most interesting results came from Guelph University, because their growth chamber had a power failure, and the temperatures increased up to 50 degrees, and these temperatures stayed like this for 10 hours. It didn't happen immediately, but 3 weeks later, the control galleys became dominant with Chlorobi OPB 56, that's a thermophilic taxon, but the inoculated plants weren't affected. And in general, all crops, both bacterial, both control and inoculated microbial communities, evolved from first 2 to 4 weeks before stabilizing, except bread wheat, as we have seen in the previous slide. Bread wheat was shifting a lot. And towards end weeks of 12th and the 6th, a similar evolution of microbial community composition occurred in the control plant. That was also accounted for bread wheat as well. But in general, the inoculated plants, microbial composition stayed more stable. With the problem with the technical failure of the Guelph University, it was also shown that the inoculated plants weren't affected because of high exposure to high temperatures. And Pseudoalteromonas, that's a very well-known PGPR in agriculture, and that was colonized very efficiently from the Myco madness. And in the second project, for ESA, at ETH Zurich, the Melissa project, we investigated the duration of nutrient deficiency changes and root shoot allometry in wheat. Melissa is the abbreviation of microecological life support system alternative, and it's a closed ecological life support system up to 70%. And we were asked to do that project because they were interested to understand the root phenotyping, the root morphology prediction under nutrient deficiency conditions, because these operating closed ecosystems with plant compartments, it needs a good prediction and a good understanding of the plant behavior under nutrient deficiency conditions. because in space, more likely a deficiency condition is expected than toxicity. Therefore, in that study, we focused on deficiency conditions, and they have been modeled the plant, the shoot growth of the plant. Therefore, they were interested to model the root morphological changes in nutrient deficiencies, and we investigated the impact of root architecture changes on the shoot growth as well. And what's root system morphology, and what does it mean phenotyping the hidden half of the plants? As you have seen in these two-dimensional pictures of the plant roots, root system morphology is the spatial arrangement, and its distribution, its proliferation style is determined by inherently by its genes and by its outside conditions. And these branching patterns is determining for the shoot system to reach out the nutrient accumulation, and plus for disease resistant, for soil compaction, and consequently this root is deciding the yield more than shoot growth actually. But because of the nature of the plant part, it's hidden under the soil, it's a little bit overlooked in plant studies. And this root foraging performance inherently related with root system morphology, but also highly changeable, highly plastic with the environment conditions. And root phenotyping is difficult when the plants are cultivated in soil, because to isolate the plant root to study without disturbing or with the intact root studies. Therefore, we deployed a hydroponic study here to investigate how temporal variability of these nutrient availability influences shoot root allometry. And our aims in this research was to quantify below and above ground responses of wheat to different durations, to 3 different durations of 7 macro and micronutrients in hydroponics of nitrogen, phosphorus, magnesium, sulfur, calcium, iron, and zinc. And we draw correlations between allometric relationships of shoot dry weight and root phenotypic traits. And we draw conclusions on how spatial and temporal variation in edaphic conditions, that's abiotic conditions, abiotic stress conditions such as nutrient deficiency, affects the plant growth. So there were 4 treatments in the experiments. The control group, the plants were cultivated till 4th week. That was an experiment till to vegetative growth phase. The control group for 4 weeks, they received an optimal nutrient solution. The week 1 plants, after the 1st week, till the 1st week, they were under deficient conditions. And week 2 to 4, in the 1st week and after 1st week, they were under deficient conditions till harvest. And week 1 to 4 plants, from the start to end, they were under deficient conditions in that experiments. And these were the most miserably looking plants, but also gave very interesting results as well. And the root system traits were obtained by using the software of Rhizo. That's a non-invasive phenotyping method of roots. It gives 2-dimensional root pictures, and estimating these root measurements of 5 parameters, which are root length, surface area, projected area, and root volume, and number of tips, and the average diameter, root diameter. These are shown and measured by rhizo shown with different colors. And we measured 30 number of root diameter classes, which means the range of wheat root diameter is from 0.05 millimeter up to 1.5 millimeter. That's a common number from literature to use 30 number of diameter classes. I will show the root phenotypic plasticity and allometric root-shoot relationships now. And allometric relationships under different durations of nutrient deficiency. On the left graph, you can see the linear regressions of root dry weight to total root surface area and to shoot dry weight. On the right figure, the shoot dry weight was correlated with the average diameter of root classes below than 0.25 millimeter and between 1 to 1.5 millimeter. And under 1 week of nutrient deficiency, the responses of shoot dry weight positively correlated with the thicker diameter, with the thicker average diameter roots of 1 to 1.5 millimeter, which is high mean root diameter. These are mainly the main roots, because main roots has the higher the larger diameter. When the plant is early stressed, it's investing on the main roots and to survive, because main roots responsible is more to uptake water and for survival. And there we assumed that the plants are prepared for stress, but we didn't catch these correlations when we look at the deficiency conditions under the 3rd and the 4th week of deficiencies. The positive correlations were captured at the shoot dry weight of the total root surface area of the thinner average diameter, that is thinner than 0.25, and these are the classes of lateral roots. And the lateral roots, they have one function in the root system to uptake the nutrients. And this high root surface area, which is investment into lateral roots in the late stress, and to keep the shoot dry weight constantly, plants get prepared for stress. When the stress conditions came upwards later. So these responses in shoot dry weight to different durations of nutrient deficiencies were related with the responses of different root traits. So actually the shoot biomass was determined by the roots and for different nutrients and also for their duration. So how can we integrate these allometric relationships into our understanding of plant growth under nutrient deficiency systems? It was important to quantify these allometric root-shoot relationships to understand the nutrient dynamics in the plant and root phenotypic root morphological traits, its plasticity, and how it is affecting the above ground plant growth and eventually yield. As said, roots are determining the yield. And as we have observed, these different root traits were determining this root-shoot biomass in short and long-term nutrient deficiencies. Although this experiment was conducted in hydroponics, but in soil, in field experiments, it's also very unpredictable environmental changes happens, such as you can do nitrogen fertilization, but the rain can come and the nitrogen can be leached out from the soil. Therefore, these the duration and the changing, the shifting, the nutrient deficiency conditions are also important to see, not for the whole course of the plant growth under one nutrient deficiency, but also in changing and situations to predict the plant growth is important. And these data is built up for a modeling process at ESA for their parameterization of their models to anticipate root and shoot growth responses to nutrient deficiencies, to different nutrient deficiencies, and the duration of this abiotic stress. And then after working at ETH Zurich for 5.5 years in lots of research projects, I started working as a food production project manager at Earth Space Technical Ecosystem Enterprises in Switzerland. ST was a Swiss startup company. I was responsible of this constructing and growing, cultivating the plant systems in that 3-floor vertical hydroponic. I mean the plant growth systems of aeroponics, hydroponics, aband flow, and LED lights, and which crops to be cultivated. That was 100% closed loop ecosystem, which means that nutrients couldn't be added. Only human waste can be recycled to be used for plant nutrition, which was a challenging task because human waste isn't designed for plant nutrition. So the idea of it was the circularity with zero waste. But the water efficiency was up to 90 degrees. There was no pesticide use, and this high water efficiency was because of the recirculating nutrients in the hydroponics. That was also in the ESA projects. The nutrients were always recycling. And the LED lights were customized, and LED lights were equipped for plant growth. And you can see the plants are standing very close by to the LED lights, because LEDs are dissipating much less heat compared to sodium vaporized lamps. And this produced heat was also dissipated and used for heating up the plant growth system chamber and also for the neighboring spaces. That was the aeroponics system, different plant species, cultivars, different LED lights. And that was the hydroponics system, different plant growth systems under the framework of hydroponics, different LEDs, in order to understand which plant growth is the most efficient for that kind of lights, for which kind of wavelengths, for which kind of light intensity, and for which crops to choose the crops. In my last position, I worked at Green City Solutions in Berlin as a moss farm manager. And these were the company products which are standing. They were called city trees. And there were 3 in Bikini Berlin, and there were 2 in Walter Benjamin Platz. And maybe one day when you visit there, you can check these city trees out. And the city tree is an outdoor air biofilter with moss. The idea is to collect up the fine air dust by the moss as a tool for mitigation of air pollution in highly polluted areas in the city, and also by the highway or facades. And it was working as an intelligent self-sensing unit, communicating online measurement data for control purposes, because the company was sending out these city trees all over Europe, and it's important still to check out the plant if something is broken, if the pump isn't working and the irrigation system was automized. And generally talking, these botanical air biofilters are hybrids of biofiltration and phytoremediation, which was first designed by NASA with potted plants. With the moss, the idea is the air is drawn through the wet biomass of the moss, and moss has a very large surface area, which is 1 square meter of moss surface area corresponds to 30 square meter of a surface area. And also when you look closely on the moss tissue, you can see the grifts on the plant. And when this contaminated air is blown through the moss, the organic hydrocarbons, the air pollutants are an ammonium. Ammonium is a pollution in the air. They are degraded by microbial film, embedded in the moss and absorbed by moss. And these organic hydrocarbons, which are human toxic, methyl ethyl ketone, toluene, benzene, all these volatile organic compounds, they are very good source for microbial communication community, the microbial film, and this fine dust gradually becomes biomass. So it's just bringing and converting these air pollutants into moss biomass, and the more biomass, the more degrading, the more breaking down these organic hydrocarbons. And that these were the most mats which were cultivated in a vertical growth system, and that was the tree. I just put it like this to see for you to help for you to understand the dimensions, the how big is the product when compared with kids. Is everyone there?
Yes.
Hello?
Yes.
Thank you. And recently now I am working as a principal investigator at Sunum at Sabancı University in a co-funded brain circulation scheme of EU Horizon 2020, Mercury Actions, and TÜBİTAK 2236. And the project is speed breeding in growth chamber to accelerate crop breeding. Climate change and slow release of new crop cultivars and global food security to feed the increasing world population is the ability to feed the increasing world population is reducing. Therefore, faster release of plant breeding, faster release of plant cultivars is needed on the market, because to breed one cultivar takes a decade, 10 to 11 years. And if a breeder is lucky enough in his whole career, maybe they can breed one cultivar. And especially the important crops of like wheat, like rice, like barley, like soybeans, like potato, which are feeding the world, more than half of the world population, they have large span of harvest times. Therefore, to shorten the generation period of the plants are needed to overcome these challenges. Speed breeding is a very new technique which was introduced like 3 years ago. It's conducted under controlled environmental conditions such as growth chambers, ecotrons, with extended photo periods, with selected monochromatic lights or mixtures of different wavelengths, and under higher light intensity when compared to a normal plant or conventional plant growth in greenhouses. Excuse me. On the right side of the you can compare the speed breeding of a wheat growth to a normal breeding of a wheat growth. In the upper part of the picture, at the day 42, the normal breeding of the wheat is not even almost close to flowering, but day 42 you can observe the flag leaf and it's almost spiking. And when speed breeding 58 day of the wheat plant, it's the spikes are visible, and in the normal breeding the plant is almost flowering. Speed breeding of long day crops, that's established, and this protocol was published in Nature Protocols. The light quality wasn't concerned. It's very fast for non-winter crops with spring cultivars. There was no arrangement on low light quality, and using regular bulbs, not even LED lights, 22 hours of light supplied to the plant, that was enough for wheat, barley, and chickpea. On field conditions, these plants in per year only 2 or 3 times of generation can be collected. For canola, on the field, only 2 times generation can be achieved. But under these speed breeding conditions, which was also conducted in the growth chamber, for wheat, barley, and chickpea, 6 times of generation was achieved, and for canola, 4 times of generation was achieved. The aim of our study, for our research, is to develop a short day protocol which allows multi-story plant growth with 5 generations per year of short day crops, because short day crops are the challenging ones, because they require less than 12 hours of light. So the challenges are short light treatment, and since the light period is much shorter than the long day crops, the photosynthesis rate is limited, and these plants are more sensible to different wavelengths, and the consequences are unstable plant stand. And the target crops are plant and potato and soybean, and customized LED lights will be used in that project regarding to be dimmable LED lights, and so you can so man can arrange the ratio of blue light to red light, and 0 to 100% dimmable. So you can just delete, or you don't use one of these wavelengths in between. For instance, between blue and red, there's orange and green, and you can exclude these wavelengths with these type of LED lights. I visited on June the Hohenheim University in the State Molecular Plant Breeding Institute, and they constructed a low-budget multi-story system. That's also what I'm going to use at SUNU. These containers are getting more popular for cheaper and for remotely controlled plant growth systems. And as they calculated, if we don't count the investment on this container, the plant growth, that was soybean, a soybean plant growth per plant, it costs 0.3 euro, including the automized irrigation system, the input, fertilizer input, the LED lights cost, the modules cost, the electricity cost, and the I think that was it. Yes. And in the project, although it will be conducted in a controlled climate chamber, temperature, the relative humidity, soil moisture, and the irrigation tank level for the automated irrigation will be controlled by microcontrollers by Arduino software. And in my last slide, I would like to introduce you that NASA astronaut Don Petit, he defines himself as a space gardener. He had a talk to us on Space for Inspiration workshop, and he showed us the zucchini plants that he carried to in International Space Station in his private box. Because he said astronauts can be able to bring a small private box, and it's not allowed to check these bags, but also it's not allowed to bring a biological material to ISS without permission in their bags. But nevertheless, he brought these zucchini seeds, and he grew it at the blind spots of the cameras that it couldn't be recorded. And he was, as also you can see here, that's the plastic plant. On the left picture, you can see the very long shoot growth. That was later development of the plant. He said the light was limiting, because since the ISS is turning, sunlight isn't always available, and the lights in ISS isn't suitable for plant growth, and the plant was always searching to reach out the sunlight, and he grew that zucchini plant in ISS. He fascinated me because working with plants also fascinates me. If you are still there, I would like to thank you again.